Have you ever thought about cell death as, well, an art form? It sounds kind of weird, right? But for our bodies to live, literally billions of our cells have to die every single day. And they don't just fall apart. No, they go through this beautiful, incredibly elegant, planned-out process of self-destruction. Okay, let's dive into this amazing process. It's called apoptosis. So that brings up a really big question. We're sort of taught to think of death as a negative, a failure of the system. But what if, at the cellular level, it's not only good, but absolutely essential for life as we know it? To really get why, we first have to understand that not all cell endings are created equal. We're talking about two very different stories here, two fundamentally different ways a cell can meet its end. It's like a fork in the road. And check this out. This diagram lays it all out for us. You see, a cell has a couple of main options. On one path, you've got necrosis, this chaotic, messy explosion. The cell just swells up and bursts. But if you follow the other path, you get apoptosis, this super neat, orderly process of breaking down into clean, manageable pieces. Two totally different endings. So let's really get into the weeds here. Necrosis is basically like a bomb going off. It's this uncontrolled, catastrophic death, usually from something like an injury. The cell swells up, bursts, and spills its guts everywhere. This causes a ton of inflammation and damages all its neighbors. But apoptosis? That's the silent assassin. It's an internal, programmed suicide that's quiet, precise, and leaves absolutely no mess behind. So why is this clean, programmed death so important? Well, it turns out apoptosis isn't just about taking out the trash. No, it's way more important than that. It's actually life's master sculptor, constantly shaping and maintaining our bodies from the very beginning. I mean, the mission of apoptosis is just, wow. It's responsible for literally sculpting our organs in the womb by trimming away the cells we don't need. It's how a tadpole loses its tail. And in our adult lives, it's working all the time, replacing billions of old cells every single day in places like our gut. And, crucially, it's our first line of defense, telling virus-infected cells or even potentially cancerous cells, hey, time to self-destruct before you cause any real trouble. And if you want to see what happens when those signals go wrong, well, just look at this. When we're developing, the skin between our fingers and toes is actually removed by apoptosis. If that process fails, if the cells don't get the memo, they stick around. And the result is webbed digits. It's such a powerful visual reminder of how essential this death process is for just building us correctly. So it all comes down to this really profound idea. This destructive process is actually one of the most creative forces in our biology. Who we are is not just about the cells that grow, but it's also about the countless cells that are purposefully and elegantly told to disappear. Okay, so if apoptosis is this super clean, orderly process, what does the evidence look like? Let's put on our detective hats, go full CSI here, and examine this cellular crime scene to see exactly how this takedown happens. One of the first pieces of evidence you'd see is a process called blubbing. As you can see, the cell's internal skeleton starts to break down, and its outer membrane begins to bulge out. It's kind of like bubbles forming in boiling water. These little bulges, or blebs, then pinch off into these tidy little packages called apoptotic bodies. Each one has a piece of the cell inside, all wrapped up and ready for easy disposal. No mess. And the next piece of evidence, Exhibit B, if you will, is in the DNA itself. When scientists analyze the DNA from a cell that died by apoptosis, they see this very distinct ladder pattern. That's because the DNA has been cut neatly into uniform pieces. Now compare that to the DNA from a necrotic cell. It's just a chaotic, random smear. That ladder is the unmistakable signature of an orderly execution. So what's the bottom line here? It's that nothing, and I mean nothing, in apoptosis is random. Every single step, from the blubbing of the membrane to the snipping of the DNA, is deliberately designed to take the cell apart without causing any collateral damage to its neighbors. It's just meticulous. Okay, so how does a cell even get the memo that it's time to go? Well, it turns out there are two main highways that lead to the same destination. Two main types of signals that tell a cell its time is up. This slide shows those two paths perfectly. Think of it like this. On one side, you've got the extrinsic pathway, which is an order coming from the outside. And on the other, you have the intrinsic pathway, which is more of an internal decision, a distress call that starts from inside the cell's own machinery. So let's look a little closer, right? The extrinsic pathway gets triggered when a molecule from the outside, often from an immune cell, literally locks onto a specific death receptor on the cell's surface. It's a direct command. You're compromised. Time to go. The intrinsic pathway, though, starts from the inside. When a cell has too much stress or DNA damage, its own mitochondria, the powerhouses, release a distress signal called cytochrome C. It's basically the cell saying, we're too damaged to function. We need to shut this down. So it doesn't matter if the order came from the outside or from the inside. Both roads lead to the same place. They lead to the activation of the final players in this whole drama, a family of proteins that are basically the cell's executioners. And these executioners have a name: caspases. They are a family of enzymes, and they are absolute masters of controlled demolition. And get this, their name actually tells you exactly how they work. The C is for cysteine, which is the amino acid they use to do the cutting, and ASP is for aspartate, the specific spot on a protein they target. They're incredibly precise surgeons of destruction. So the caspase team is basically split into two groups. First, you've got your initiators, like caspase 8 and 9. Their job is to get the first signal from either that extrinsic or intrinsic pathway. Once they get the signal, they turn around and activate the second group, the executioners, like the famous caspase 3. And these are the ones that go out and methodically chop up all the important stuff, taking the cell apart from the inside out. And this right here, this is the crucial part. This is the point of no return. Both the outside-in signal and the inside-out signal ultimately have one goal: light the fuse on those executioner caspases. Once that switch is flipped and these enzymes are unleashed, the cell's fate is sealed. The process is totally irreversible. And that brings us to one last really critical thought. If this art of cell suicide is one of our body's greatest protections, what happens when a cell breaks the rules? What happens when a rogue cell, like a cancer cell, figures out how to switch off this machinery and just ignore the order to die? Well, that's when this beautiful process of life and death becomes a matter of life or death for the whole organism.